Hi everyone, in this video I'm going to show you using this very tech-savvy model how DNA is replicated inside a cell. So just for a moment of clarity, get your mind back in context. We are thinking about the cell cycle, synthesis phase. All of the DNA in the cell needs to be replicated so that the cell can undergo mitosis later on. So we're talking about meters and meters of DNA, but for the purposes of this, we'll just stick with these 8 bases that we have in front of us. So the first thing that's going to happen in order to replicate this strand of DNA is we need an enzyme. See how fancy this is. The enzyme is called DNA helicase, and it's a bit like a pair of scissors. And what it does is it zips down, chop, 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 and it cuts through the hydrogen bonds. You can see they've broken already. It cuts through the hydrogen bonds that hold together the 2 strands of DNA. So once that happens, we end up with 2 completely separated strands. But notice that their sugar phosphate backbone, their phosphodiester bonds are still intact. So they're still 2 strands. They're just not joined together anymore. Now inside the nucleus of the cell, there are a whole bunch of just free-floating nucleotides that aren't connected to anything. And so what will happen is, because of the nature of complementary base pairing, we will get these bases sort of lining up, and they will automatically form pairs with their complementary partner. And what will happen here, which I might have to put you down to show you, what will happen here is all of these hydrogen bonds will reform by themselves, because hydrogen bonds, as you know from water, form spontaneously. They just happen all by themselves. So these, the 2 strands will actually be attached to each other in the middle. Bear with me one second. Okay, so I've just spontaneously formed the hydrogen bond. So we've now got 2 strands of DNA. They're stuck together here and here. But as you might have noticed, their phosphodiester bonds are significantly... Oh, I've lost my hydrogen bonds. Their phosphodiester bonds are significantly lacking. So what we've got is we've got a solid chain on this side with a sugar phosphate backbone. But the new strand is just these loose nucleotides. And so what we need to do is need to get another enzyme. This enzyme is called DNA polymerase, and it's a little bit like a zipper, or you can imagine it like sticky tape. And what it does is it forms the bonds between the deoxyribose sugar of one nucleotide and the phosphate of the next nucleotide. So I'm not going to do this, we'll be here all day. But yeah, so it joins the phosphodiester bonds together. So we end up with 2 complete molecules of DNA from 1. Now what you'll notice about these is that 1 strand in each is old and 1 strand is new, and here we've got 1 new strand and 1 old strand. And so for this reason, the mode of replication is called semi-conservative, because it partly conserves the old DNA and it partly generates it new by using a template strand. Okay, so semi-conservative means half old and half new, which you can see I've represented by the use of the green pen and the black pen. I hope that makes sense.